Good morning, everybody. It's kind of dim in here. I'm just going to put a light on. Not that you need to see me any better, to be honest, but there you go. I'm in all my glory. Okay, good morning, everybody. Happy Friday. Woo. Not too long before we're done, which I'm sure everybody will be quite happy about. So today we have, we finished up, where at? Let's go on computer. Computer thinks it's Friday too. Okay, get the chat up. I don't know why everything's going so slow. Just give me a moment. Otherwise, this is going to be kind of torturous if I can't figure out what's going on. There you go. That's better. All right. So we left off talking about, we did talk about the catabolite activator protein and cyclic AMP, right? You need to remind me because I often get confused between. Dang it, where's that? Here it is. All right, I'll go look myself. Yeah, we did. There we go. Cool. Oh, well, thank you, Johnny. I appreciate that. So that's where we ended up. Should have done that before class. I just woke up late, to be honest. Kind of pooped, to be honest. So just to recap the lac operon. The lac operon is inducible, right? So inducible means that expression is normally off. There's Caramel Cat being a little pest as usual. Got fur flying in front of my face. And the inducer is lactose, right? Because there's no point expressing the, I'll get you back. There we go. There's no point expressing the lac operon if there's no lactose around to metabolize. You know, it would be a futile and wasted effort. So lactose and the removal of lac-I, which is the repressor of the lac operon, allows huge amounts of expression from the lac operon. And the fine tuning, right? You know, I kind of talk about this a lot. You know, you got the big on-off binary switch, lactose present or not. And then you got the fine tuning based on how much glucose is around, right? Because if there's a lot of glucose around, you don't need to turn lactose into glucose. And that's done using the catabolite activator protein as a sensor to detect levels of cyclic AMP. And cyclic AMP levels are inversely correlated to glucose levels. So the more glucose you have, the less CAMP you have, the less likely you're going to have the CAP-CAMP complex, CCC, aiding RNA polymerase binding, right? And so transcription levels drop. Now the tripoperon is serious business, right? So in some ways the tripoperon is simpler because really we don't have two different conditions that we care about here. So in the lac-operon is lactose present or absent? Is glucose present or absent, and at what level? Right, those two things have to be kind of figured out essentially to result in a given level of lac-operon expression. So the tripoperon is a lot simpler. So it's all about the production of tryptophan, right? And so tryptophan is an amino acid. There we are. Right? And it's produced from, at the beginning of that, compound called chorosmate, whatever that is. So this is a substrate. The tripoperon encodes all the genes TRPE, D, C, B, and A that are necessary to produce the enzymes that turn chorosmate into tryptophan. Okay. And so once you have enough tryptophan, then you don't need as much tripoperon expression, right? So this is a great example of a feedback loop. So as tryptophan levels rise, the amount of tripoperon expression decreases. So essentially, if you're a bacterium and you're doing a lot of gene expression and translation, you're going to need tryptophan, and tryptophan will always be being consumed, right? So tryptophan's an amino acid. It gets stuck on the end of the tryptophan tRNA, and then that gets used to make proteins, right? That's a very, very important point, right? So anytime I say that's a very important point, make notes of that because it's really important. That's what I'm telling you. So tryptophan is always being produced, right? So this is a system which is on normally. It's the opposite of the lac-operon, which is normally off. It makes sense. So if you think about the context, you only want to produce products from the lac-operon when lactose is present. So it should be off when it's not needed. You always are going to need tryptophan at some level or other because it's an amino acid. It's not an essential amino acid. That means something different, right? So the tripoperon is normally on, but it can be turned off. So therefore, the tripoperon is repressible, right? And it's a good example of a feedback loop where tryptophan levels, once they reach a certain level, act to inhibit the transcription or repress the transcription of the tripoperon. Okay, want to make any questions you have at any point. This one is something which I pretty much guarantee you're not going to get first pass. Not because you're not capable, but because it's complex and there's a lot of moving parts. But also this is an astonishingly elegant way of controlling transcription. Not just what I'm showing you now, but the bits I'll be showing you in a little bit. Correct. Generally, when tryptophan levels are high, the tripoperon is off. Okay, so this mechanism here is the binary switch, right? So remember we talked about the lac-operon, it's either on or off, right? Tryptophan levels, once they reach a certain level, like really high, is the same deal, off, right? Or whichever way you pull it, don't know, doesn't matter. So the high levels of tryptophan are the binary switch in the tripoperon. And because the tryptophan is the product of the things produced by the trip operon, this is a feedback loop, right? It's a negative feedback loop because as the product of what the things produce increases, it decreases expression. So this is the relatively simple part, right? We have a repressor called TRPR, and this is an aporepressor. So that means it needs a corepressor to bind to the TRPO operator. Dang, can't see what I'm writing here. And that corepressor is tryptophan. So TRPR and TRP equals repressor. And the reason being is the presence of tryptophan changes the shape of the TRPR protein so that it can bind DNA. And that TRPR protein is produced from a gene elsewhere in the bacterial genome. So when that TRPR tryptophan complex is bound to the operator, RNA polymerase cannot bind, or I'm not entirely, it varies. It can either not bind or it can't process through the operator. Either way, it doesn't function. Everybody got that? Cool. That's the easy bit. So there's lots of different ways I can teach you this. Let's try in a different way with the other class. So let's see if it helps with this one too. Okay, so what's one of the key characteristics of gene expression in bacteria? There are a few. What can you think of? Okay, what are some of the key characteristics of gene expression in prokaryotes? Correct. It's usually negative regulation, so it's usually genes are normally on and they're turned off. That's good. What other, think about the whole picture, not just the control of transcription, but the whole shindig. Also think about what the bacterial cell is like compared to a eukaryotic cell. So what's one of the easiest ways to define a eukaryote over a prokaryote? What's one of the major differences? What does a eukaryote have that a prokaryote doesn't? There's many compartments. Good, Jelly. Excellent. Right? So if DNA is in one compartment and ribosomes in another compartment, what can you say about transcription and translation in eukaryotes? They have to be separate, right? So one of the big differences in prokaryotes is that transcription and translation are coupled. So that means that as soon as RNA polymerase kind of moves down the DNA and it has a bit of mRNA sticking out, a ribosome assembles on that and essentially translates the mRNA as the RNA polymerase is producing the mRNA from DNA. Right, so those two, as soon as the RNA polymerase, as soon as it's basically moved kind of onto any region that has a Shine-Dalgarno sequence before it, which is what allows the prokaryotic ribosome to assemble. As soon as that RNA polymerase has moved past that and that Shine-Dalgarno sequence is flapping in the breeze, a ribosome will assemble to it and it will move along in lockstep, right? And then another ribosome will assemble and then another one, and you'll get the polypeptide chains being produced before the mRNA is finished, right? So those two processes, and this is why I mentioned it a bunch earlier on in the previous section, are linked, and that is absolutely, completely, totally important, very important, for how the tryptophan operon is fine controlled. So the fine control of the trip operon is called attenuation. And so attenuate means to cut something short, right? You know, if I was to go, oh, I'm too tired, don't want to teach genetics no more today, it's a Friday, I'm going to attenuate the class. That'd be a super fancy way of saying I'm stopping the class, right? Early. So we've got the extremes, no tryptophan, lots of tryptophan. Put my hands a little bit closer together so you can see them, right? In between that, we need to fine tune the levels of trip operon expression. And the reason is that the use of tryptophan in making proteins is not constant. Some proteins need more than others. Bacteria go through periods of not doing very much to periods of doing lots of, gene expression and protein production. So the need for tryptophan is going to vary constantly. And so the production of tryptophan also needs to be varied constantly, right? And so most of the control, if you think about it in a general everyday, day-to-day kind of context for a bacterium, if they even think about it in that way, hey, they don't think, they don't have brains, they're just bacteria. But has to be somewhere between those two extremes, right? It'd be kind of crazy that the bacteria has no tryptophan, right? Because that would mean that it basically is either going gonzo on translation or something badly wrong. And there are a few situations such as, the bacterium going into stasis or something like that, where you're not going to have any translation occurring, right? So you'll have a load of tryptophan. Day-to-day biology, essentially, is going to be somewhere in the middle. So how can you detect tryptophan levels? That's the key. So you can detect if it's really high using the TRPR aporepressor. But we're not worried too much about that because that's kind of a slightly odd situation to be in. How do you detect intermediate levels of tryptophan, right, between nothing and a lot? And so that comes back down to what tryptophan is, right? So we're not going to be sensing this indirectly. Well, no, we're not really. Like we do with glucose, right? We're not going to use a proxy, right, which is cyclic-AMP in the case of glucose. We're going to detect the levels of tryptophan directly. So what is tryptophan? It's an amino acid. What is tryptophan added to before it can be used in translation? What molecule? Talked about this a bunch in terms of translation. What molecule in the cell will tryptophan amino acid be added to? Yeah, that's good. But before it can be added to a protein, what other molecule must it be added to? So it can enter the ribosome. So you're right, eventually it'll be part of, yeah, that's just another way of saying protein. Before it can be added to a polypeptide chain. So what enters the ribosome at the A site? Almost. mRNA is the template. It's not what's used to make protein. Well, it kind of is, but not directly. There's another type of RNA. You're on the right track. What type of RNA do amino acids get added to? Just pick a different letter. tRNA. Yeah, thank you, as I said. No, that's exactly right. Okay, so if tryptophan is present, it's going to get added to a tRNA, right? So to detect tryptophan levels, we need to basically see whether we can incorporate tryptophan into a polypeptide. That's the most direct way of doing so, right? And this is where things get really complicated. So I'm going to take my time and I want you to ask any questions you can think of and stop me any point stuff doesn't make sense. Okay, this is really important because this is a very difficult thing to get first time round. I know it certainly was for me when I taught this course first in 2014. Yeah, that's right. First full-length course I ever taught. It is an absolute disaster. Anyway, so thankfully I've moved on a bit from then, as one would hope. So this is a really difficult concept. So we've got coupled transcription and translation. Okay. We've got the product of the trip operon, essentially, is involved in translation. And so we're going to sense its levels by seeing whether or not tryptophan can be incorporated into a protein. Okay. And whether it can or cannot is going to determine whether or not the RNA polymerase transcribes the trip operon. Okay. So we've got a bunch of steps here, and I'll kind of write them out so that we have them up in front of us. So we're going to detect trip levels by seeing whether or not a tRNA trip is present for translation. And this ties into the coupling of transcription and translation in prokaryotes. Okay, so basically, if tRNA trip is present, equals small, oops, can't spell small, come on. small protein is formed, or translated rather, that's a better word. Equals no RNA pol transcription. If no or little tRNA trip is present, equals stalled translation of polypeptide, equals RNA pol transcription. That's the basic mechanism of attenuation. And so you're probably wondering, okay, well, you know, what is this polypeptide, right? What are you talking about? What's going on? And so what we're looking at is a short stretch of sequence between the trip operator and the first structural gene, which is TRPE. This is called the leader region. So again, remember, TRPO is where the TRPR and TRP repressor protein binds to. So if tryptophan is present in really high levels, then the TRP protein will bind here, and RNA polymerase will not be able to transcribe these structural genes. Okay. Now, if tryptophan is not present at super high levels, TRPR does not bind. RNA polymerase can transcribe this mRNA potentially. And so this leader region consists of a small bit of mRNA that encodes a small, what I would call dummy polypeptide. This polypeptide isn't used for anything other than the regulation of the TRP operon. Right? It doesn't go on to do any metabolic process. In fact, as soon as it's produced, it gets broken down, and those amino acids get used again. Right? So this small polypeptide encoded in this leader region is purely there as a mechanism to control the fine control of expression of the TRP operon. Okay. Anybody got any questions so far? Okay, so the next step that you need to get is, how does transcription get terminated in prokaryotes? This is going back a way now. What are the two main mechanisms for transcript or transcription termination in prokaryotes? Anybody remember? I'll give you one. One was row-dependent or extrinsic termination, relying on a protein. Anybody remember the other one? Intrinsic. Good, thank you, Jeremy. So intrinsic transcription termination relies on the formation of what? If you have long hair, or if you've just cut it and you can't pull it back in a ponytail, you often use these things to hold like stray strands in place. What RNA structure forms? Yeah, awesome, a hairpin loop, exactly. Right, and the hairpin loop punts RNA polymerase off of the DNA. So, guess what we have in this leader region? We have a possible hairpin loop terminator sequence. Now, this is a possible one. It doesn't always form. The formation of that terminator loop depends on whether or not that dummy polypeptide is translated. Okay, so now we're starting to see, how this mechanism can work, right? So we're kind of building up, right? That's why I wanted to try this, see if it works better than other ways I've taught it in the past. We're kind of building up these concepts, basically starting with the coupling of transcription and translation, right? And now we have two things. We have a dummy polypeptide, which may or may not be translated depending on how much tRNA with tryptophan is present. And we have a terminator sequence, which may or may not be formed depending on whether or not that protein is or polypeptide is translated. Oh, also forgot. Also, in the, I feel a little bit bad calling it a dummy polypeptide because it's, I don't know, I feel it seems a little bit pejorative or derogatory perhaps. mRNA are two trip codons. Okay, so now we're getting somewhere, right? Now we have the requirement for two charged trip tRNAs or tRNA trip or tRNA trips. Trip tRNA sounds a little bit easier to say. For that polypeptide to be translated. And so essentially, we have a conditional system here. If tryptophan is present, because it's being produced as a result of expression of the trip operon, then this leader region gets translated because tryptophan is available to get added to tRNAs. Those tRNAs come in when those two trip codons, need to be translated. And in doing so, we have the formation of a terminator loop or terminator hairpin loop, this attenuator sequence here. Right, so and we'll go through the mechanism of how that works in a second. So if the trip tRNA is present, the dummy polypeptide is translated, equals terminator loop forms, equals termination of transcription. If tRNA trip is not present, or not present at a high enough level that it's reliably incorporated into the polypeptide, dummy polypeptide there's a P there, hang on. So it's not translated equals no terminator loop formation equals continuation of transcription. Okay, I'm going to give you a few minutes to cogitate on that, see if that makes sense. And again, if it doesn't, that's totally cool. Just ask questions and I'll help. Any questions? All right, so, sorry. Okay, so this all moves around this idea of RNA secondary structures, loop formations, right? So RNA is not typically a linear molecule of one strand, although it's often portrayed that way. RNA is actually a fairly complex molecule that can take all kinds of shapes depending on context. And so this is all about, pardon me, this is the leader sequence here. Okay, so this is basically this is like well, it's not really a 5' UTR because it is translated partially. But we have this, let's move this out of the way so I can see what I'm talking about. So this is the start of the TRPE gene here, and this whole thing here is what's called the leader sequence. Okay, and this region here, where those U's are, remember, because U's cause RNA polymerase to stall or to slow down. These regions here are what can form the terminator loop. But essentially, we have 4 different regions in this leader sequence, and they can pair with each other in different combinations. Right? We can get 1 and 2 paired together, and then that will allow 3 and 4 to pair together. 3 and 4 paired together are the terminator loop. Right? That's essentially RNA polymerase will be around here, and if that terminator loop forms, it will kick RNA polymerase off. Also, loops 2 and 3 can form a loop as well, right? So if loops 2 and 3 form a loop, 3 is no longer available to pair with 4, so you don't get the terminator loop formation, right? That's how this all works. And the thing that controls which combinations form is whether or not the ribosome pauses over these 2 TRP codons. And remember, RNA polymerase is kind of around here as the ribosome is over here, right? So as soon as that region 1 is cleared by the RNA polymerase, it can be translated, right, by the ribosome. So essentially, if tryptophan is present, tRNA is available for adding tryptophan where these TRP codons are. And that ribosome will move over into this region here, right? So it will cover 1 and 2. Does that make sense? And so if 1 and 2 are covered, RNA polymerase is going to produce the mRNA, which can then form where 3 and 4 can pair, right? Because 4 is produced after 3. And that will form a terminator loop, which will terminate transcription. The reason why 3 isn't pairing with 2 is because the ribosome is covering 2. It doesn't physically allow it. Does that make sense? Because it's already moved along after translating the trip codons in region 1. So now we get some more conditional stuff, right? If the tRNA trip is present, let's move this over a little bit. Region 1 is translated and the ribosome moves onto region 2, preventing it from forming a loop with region 3 equals region 3 and 4 form the terminator from the termination loop equals termination of transcription. Now, if tryptophan levels are low, right, the ribosome pauses, right, over region 1 because there's no, it's almost like these are stop codons, right? They're not, because the release factor doesn't come in. But effectively, it stops translation because there's no tRNA coming in for that polypeptide to get extended and then the ribosome to move along the RNA, the mRNA. So if that happens, region 2 is going to be free. And as soon as region 3 is produced by the RNA polymerase, it forms a hairpin loop between 2 and 3. Now, this isn't close enough to the RNA polymerase, and the RNA polymerase is not paused around region 3 because there's no poly U sequence. Right? The RNA polymerase has to be paused over those poly U's to be able to be kicked off by a hairpin loop. So if region 2 and 3 form a hairpin loop, there is no terminator loop formation and transcription proceeds. So if tRNA trip is not present, ribosome stalls over region 1, which allows region 2 and 3 to form a loop. And the reason why it isn't between 3 and 4 is because region 2 and 3 are produced before 4 is. And so there is no 4 for 3 to base pair with because it hasn't been made yet. So as soon as 3 is produced, essentially it has the option of, well, if 2 is available, it will base pair and form a loop with 2. So you'll have a 2-3 loop. If 2 is not available, it will just hang around until 4 is made, and then it will form the terminator loop. right? So if 2 is free and available, it will form another loop, which is not a terminator loop. So basically, you get no 3-4 terminator loop equals continuation of transcription into the structural genes. And that's essentially shown really quite nicely here in a sequence of events, right? So this is a really nice diagram, which I recommend you spend some time going through, right? It essentially shows the two things together: RNA polymerase here and the ribosome, right? So as soon as the RNA polymerase has moved far enough downstream, 80 base pairs or so, the ribosome is able to bind and start translating region 1. Now, if there isn't a lot of tryptophan around, you can see the polypeptide coming out here. This line at the top is the mRNA. This double-stranded line at the bottom is the DNA. And so essentially, this ribosome pauses because there isn't any tRNA with tryptophan attached to it to incorporate into this polypeptide. Region 2 is produced. Already RNA polymerase moved on. RNA polymerase will then move on and transcribe region 3 before it transcribes region 4. And so that will allow 2 and 3 to form a loop. And if 3 is already bound to 2, it can't form a loop with 4. Now on the flip side, if there is plenty of tRNA with tryptophan around, or enough for this ribosome not to stall, right? Again, that's why there's 2 trip codons in region 1. The ribosome processes onto region 2. So region 3 is produced, but there's no region 2 for it to bind with because it's already been occupied by the ribosome. And so the region 4, when it's produced, is able to form this 3-4 terminator loop, and that terminates transcription. And that's all because transcription and translation are coupled in prokaryotes. That's what allows this whole mechanism to work. Hello, girl. Professor Luna in the house. She's actually just waiting for me to let her outside to go to the toilet. Okay, now this is going to be a lot to take in one go, and I know that. Yeah, she's very cute. So don't expect yourselves to understand this first pass. I'd strongly suggest spending time going through this, particularly these last 3 slides, because attenuation is the hardest part of this to get. But it's also the coolest part because this is a really elegant way of detecting something, right? You think how hard it is to detect, like, what does an amino acid look like, right? How do I detect that? I don't have any eyes, right? This is a really, really cool way of detecting the level of amino acid by whether or not it can be incorporated into a protein. So I think that's pretty cool. Okay, I think that's enough for now. Yeah, it is tough. I mean, that's the thing about genetics, is that everybody has a different strength and weakness in genetics. And most people do better with the inheritance type genetics. Even though it's math-based, it's fairly straightforward logic, right? And the molecular biology aspect, people generally struggle with because it's about a lot of details, right? And those details are all really, really important. And so that's where you can get like the general idea, but if you don't get the details, then it just doesn't work at all. So that's why these last two sections are a lot tougher, right? Because there's a lot more detail, a lot more kind of complex concepts involved. That's also why this stuff is so interesting too, because it is the molecular basis of the bigger picture stuff that we learn at the start of the semester. So you kind of can't have the benefits of understanding cool stuff without essentially the work of understanding the cool stuff. Anyway, that's enough from me for today. I hope you all have a lovely weekend. Keep studying hard, please, because exam 4 will be as tough as exam 3 was, right? And so I want you to be as prepared as you can be for that, okay? All right, and bring any questions with you to class on Monday. Exam 4 is, give me a second, Jeremy. Doing exactly what? Exam 4 will be on the 30th of April. So 2 weeks time. Cool. All right. Catch you later, everybody.